comes science. They might be trying to here comes science.
Hi, Dr. H here. Today's video will be our Unit 2 review, where we will be getting into cells and membranes. This material, I believe, will be a little bit more likely to show up than the Unit 1 material that we covered in the last video, but it's hard to say what is actually going to be on the test. So let's jump right in and talk about some cells. One of the first things that we went over with cells is the fact that they are very, very small. Even in very large multicellular organisms, cells are still very small. And the reason for that is they have to efficiently exchange material with the environment. And this is best done with very small volumes. One thing I would like to point out here, this set of formulas will be provided to you on the formula sheet with your test. I don't know that you will actually have to use any of them. These are all the surface area and volume formulas for all types of three-dimensional shapes. But I did just want to point out that they will be given to you. And remember, when we are looking at cell size, the important aspect is this surface area to volume ratio. Right? We want that ratio to be as high as possible. The other big aspect of basic cell structure that we looked at was the idea of compartmentalization, that cells are divided out into different areas for different functions. And this is true down to the very, very simple bacterial cells. Right? They have discrete areas for discrete functions. And certainly when we move into the more complex eukaryotic cells, we certainly see here lots and lots of compartmentalization with all of our internal membranes and all of our organelles, which was, of course, the next topic that we got into. And these are, of course, our specific cell structures that perform specific roles. Out of this whole list that we see over on the side, the only one that is present in all cells, and that would be bacteria, archaea, and eukaryotes, are the ribosomes, right? The organelle that creates proteins. Okay. I am not going to go through all of these organelles in this video because I feel like we had should have a pretty good understanding of what all of these organelles do. But if you do need a quick refresher, make sure that you go and look up any specific organelles that you may need to review. Okay, one topic that I am going to touch on a little bit here with organelles is this endosymbiotic theory. And this theory relates back to the early formation of our energy organelles, the chloroplast and the mitochondrion. And where did these come from through evolution? So the idea behind the endosymbiotic theory is that these organelles were once free living organisms that were brought into an existing cell, and instead of being digested for food, for energy, they actually ended up living inside of this cell and evolving to become this dependent organelle. So the idea here, as shown here, there is an existing eukaryotic ancestor, and this oxygen-using early cell is brought in, and it then over billions of years evolves into the mitochondrion, and then sometime later this occurs again with some early photosynthetic organism, and that then evolved into the chloroplast. So there are a number of lines of evidence that we have to support this. First off, the double membrane structure of these two organelles, where the inner membrane would represent the original cell membrane, and then the outer membrane would have come from the engulfing cell as it formed a vacuole around this this incoming cell. Another good piece of evidence is that each of these organelles, mitochondrion and chloroplast, both have their own DNA. And a lot of the proteins involved in respiration in the mitochondrion and photosynthesis in the chloroplast, these genes are actually coded by that DNA within the organelle. A final third line of evidence that we have to support this endosymbiotic theory is the way these organelles divide. Okay, they divide on their own cycle, not with the overall cell cycle where we see the cell dividing, and the way that they divide very closely mimics the type of cell division that we see now in bacteria, right, called binary fission. Okay, so those three lines of evidence very strongly support this endosymbiotic theory that the mitochondrion and the chloroplast were once free living organisms that were engulfed in this early eukaryotic cell and took up residence, and over millions or maybe billions of years of evolution have developed into these dependent organelles. Okay, so that pretty much wraps up the cell side of the unit. So now let's jump over and look at some membranes. Cell membranes, or plasma membranes as they are sometimes referred to. These are present in all cells. Okay, they form the boundary of the inside of the cell and the outside of the cell. And as I mentioned earlier, eukaryotes all have internal membranes to help with compartmentalization. Looking at the structure here, and we'll get a little bit more into the structure in a minute, they are the main model of cells of membrane structure is the fluid mosaic model, where the fluid portion of the model is the phospholipids and the mosaic portion is all of these proteins that are associated with the membrane. Okay, so looking a little bit closer at the structure, I've already mentioned our fluid mosaic model, right, made up of phospholipids and proteins. So looking at the phospholipid side of this, remember that phospholipids have the two fatty acid tails and a polar head group. So the fatty acid tails would be the non-polar region, that would be hydrophobic, and the head region, the polar head, would be the hydrophilic region. So because this molecule has these two very different properties, right, and we call that an amphipathic molecule, when we mix phospholipids with water, they will spontaneously form this bilayer structure, okay, with two layers of phospholipids with the heads facing out towards the water and the tails sandwiched in between with all of the water removed, because remember, the tails, being hydrophobic, do not want to associate with water, right? These hydrocarbon tails are all non-polar, right? Very hydrophobic. One additional important feature of these phospholipids is whether or not the fatty acids are saturated or unsaturated. Right? Remember from the last video in Unit 1, unsaturated fats have carbon-carbon double bonds within the tails. So that introduces a bend in our fatty acid, and what happens is that the phospholipids cannot pack as closely together when the tails have this double bond. If the fatty acids are saturated and they are all carbon-carbon single bonds, then the tails can pack nice and tight together. So this is very important for organisms that have evolved to live at colder temperatures. If an organism lives at a cold temperature, and all of its fatty acids in its cell membrane are all saturated. They'll pack very, very tightly together, and the membrane will turn solid. Okay? Cells cannot survive with a solid cell membrane. So the organism will introduce these double bonded, these unsaturated fatty acids into the cell membrane, and that helps it adapt to colder climates. Okay, as these unsaturated fatty acids become more prevalent in the cell membrane, the membrane will stay fluid at lower and lower temperatures. Okay, so this unsaturated versus saturated fatty acid content is very important to keep the membrane fluid at low temperatures. Looking structurally really quick at the protein content of the membrane, a lot of these membrane proteins will go completely through the cell membrane, so they will be exposed on both sides, right? The extracellular side, that would be the outside, and then the cytoplasmic side, that's inside the cell. So the surface characteristics of these proteins have to change based on where they are in the membrane. So the protein surface that is exposed to the extracellular side and the cytoplasmic side, that those amino acids would all be polar or hydrophilic because they are exposed to the aqueous solutions. The amino acids that are embedded within the protein inside where all the fatty acid tails are, those will all be nonpolar or hydrophobic amino acids because those are going to associate well with that region of the membrane. So mutations that maybe change the protein, change the amino acid properties, so could change the way the protein fits in the membrane, could make the protein not work if it cannot embed itself into the membrane properly. Okay, so looking now at membrane function. Okay, the main function that we are going to be talking about here with membranes is in transport. And the phospholipid portion of the membrane is really what controls the transport and regulates what can pass through the membrane and what cannot. So small non-polar molecules can generally pass right through the membrane. Things like dissolved gases like oxygen can move through the membrane very, very easily because they can associate with that non-polar region in the middle. Okay. Molecules that are not able to easily pass through the phospholipids, so very large molecules or charged polar molecules, are going to need a little bit of help, and one of the many roles of the membrane proteins is with transport. It will bring in these molecules that maybe cannot pass through the membranes easily themselves. Okay, and here we see a number of other protein functions of these membrane proteins. Not really going to get into too many of them here. We will talk a little bit about cell-to-cell recognition and signal transduction in our communication unit review. But for right now, we are really going to focus on the transport properties. Okay, so transport. Substances, when left on their own, substances will diffuse. This means that they will move from an area of high concentration to an area of low concentration. Okay, this is just simple diffusion. We don't need a cell to see this. We just need two areas of different concentration. And here, in this example, we see that we have this selectively permeable membrane, kind of mimicking the cell membrane, and the large solute here cannot move through this membrane, but the water molecules can. So by adding this solute, we are basically lowering the concentration of water, so it is going to move in a certain direction. So different types of diffusion that we may see. Simple diffusion, that's one that I have already mentioned. This is molecules that are able to move through the membrane themselves. So these are the small non-polar membranes, like our dissolved gases. Facilitated diffusion, a few different types of that pictured here. This is when a larger molecule or a charged molecule that would not be able to pass through the membrane itself through simple diffusion needs some help from a protein. So a carrier protein here shown helping glucose move through will have a little bit of a shape change as the glucose comes in, and our channel protein here moving our charged chlorine ion through, that just forms a channel for the chlorine to move right through. So when we talk about transport and we look at transport, we have two different types, passive transport and active transport. So we'll start here with passive transport, and this is substances moving down a concentration gradient, right? Moving from an area of high concentration to an area of low concentration. And this is just due to the random movement of the molecules. And once the situation gets to equilibrium, once our system reaches equilibrium where there is equal concentration, then the net diffusion will stop. But remember that the overall movement of molecules does not stop. Just we don't see a net movement from one area to another once we are at equilibrium. So looking at this from a cellular aspect, with passive transport, there is no energy input from the cell. Right? This is just substances moving down the concentration gradient. So that's an area of high concentration to an area of low concentration. Types of passive transport. Again, we have our simple diffusion. That is just molecules moving right through the membrane. And then we have our two types of facilitated diffusion, either moving through a channel protein or being moved by a carrier protein. These two types of facilitated diffusion are very, very similar. Just does the protein stay in one shape, or is there a little bit of a shape change with our carrier protein? Again, the important thing to remember here, there is no energy input from the cell. One other aspect that we should mention here with passive transport is tonicity. Right? What happens when we put cells into different concentrations of solutions? So hypotonic, isotonic, and hypertonic. Right? These all are relative terms. In this case, they are being used to describe the surrounding solution with respect to the internal cytoplasm. Hypotonic means that the surrounding solution has a lower concentration of solute. So what's going to happen in an animal cell? The water is going to be moving into the cell is going to swell and potentially burst. In a plant cell that has a cell wall, water will be moving in, and this is the natural state for a plant cell, and it's going to have a high level of turgor pressure, which is what the plant wants in order to hold itself up. In an isotonic state, this means that the solute concentration is equal inside and outside of the cell. So as shown here in the animal cell, we are at equilibrium. There is an equal movement of water into and out of the cell. So this is where an animal cell wants to be. An animal cell wants to be in an isotonic situation. A plant cell in an isotonic situation is going to lose its turgor pressure, and this could potentially cause the plant to wilt, right? The plant cells will go flaccid. Okay, and our final situation here, the hypertonic state, this is when the solute concentration outside is higher than what we see inside the cell. So this is going to cause water to come out of the cell, and in both of these cases, in an animal cell and in a plant cell, we are going to see the cell shrivel up, and this could potentially lead to the death of the cell. There is another set of formulas here that will, again, these are pulled right from our formula sheet, and these have to do with water potential. And this is really used mainly for plants and trying to determine which way water is going to move, right, into a cell, out of a cell, from one area of the plant to the other. So again, I don't know if these formulas are going to be used on the test, but they will be given to you. So I did just want to show them to you again, where water potential, represented as psi, is the sum of the pressure potential and the solute potential. And we can calculate solute potential by using the second formula here. Psi s is equal to negative I CRT, and all of those variables are given to you here. So we may see these formulas on the test, but just be familiar with them. Shouldn't be that hard to use. Okay, so that wraps up passive transport. Now we will talk about active transport, and this is the movement of substances against the concentration gradient. So this is when things are moving from an area of low concentration to an area of high concentration. And from a cellular standpoint, this is going to require energy input from the cell. And that energy, of course, will be in the form of ATP. So our best example of active transport that is used by all the cells, which is constantly running, is the sodium-potassium pump. Okay, this pump is, like I said, is constantly working, and it pumps 3 sodium ions out of the cell with the use of ATP, and it will bring 2 potassium ions into the cell. This pump is very, very important. We will talk about this pump again when we get into the nervous system and start talking about how neurons function. But remember, this is moving substances against the concentration gradient, really setting up a concentration gradient, and it requires the input of energy in the form of ATP from the cell. Another important aspect of active transport is this idea of co-transport. Okay, and this sort of goes back to what I was saying about the sodium-potassium pump, where we are setting up this concentration gradient. And it's important to remember that whenever we have a concentration gradient, whenever we have a difference in concentration on 2 sides of a membrane, there is energy within that gradient. So this co-transport uses this energy to transport a second molecule. So here we have our active transport of protons, right, through our proton pump. So that is using ATP to pump these protons from the cytoplasm out to the extracellular fluid outside of the cell, and that sets up this hydrogen ion gradient where we are high outside the cell and low inside the cell. So once we have this gradient, there is energy within that gradient. So this co-transport can then use that energy to bring something else into the cell. In this case, we are bringing in sucrose, a disaccharide. Very large molecule, it's highly polar, so it is not going to get into the cell by itself, but as this co-transporter allows hydrogens to flow back into the cell, flowing down the concentration gradient, this is going to release the energy within the gradient, and that energy that's released can then be used to bring the sucrose into the cell along with it. Okay, so the direct use of energy here is to build up, to create this hydrogen ion gradient, and then the energy within that gradient is then used to bring sucrose into the cell along with the hydrogens coming down the gradient and releasing that energy. That is Unit 2, Cells and Membranes. I hope that all made sense, and I hope to see you guys soon.
Dropping science like Galileo dropped the orange.